Hi, I'm Sam Imberger from the University of Melbourne, and today I'm going to give you a bit of an update on Project B1, which is also known as Urban Flow Ecology. So, investigating relationships between flow, channel form, in-stream vegetation, and ecosystem function. So, this is quite a new project, and at the Melbourne University end, it involves myself, Joe Greet, Matt Burns, and also Scott McKendrick. Okay, so research has shown that urbanisation drives increases in peak flow magnitude, frequency, and geomorphically effective flows. And we know that this increases coarse sediment export, it reduces organic matter storage, and it also reduces the diversity and abundance of in-stream vegetation. However, we currently lack an understanding of which components of the flow regime are most significant at influencing sediment and organic matter dynamics and how they interact to influence in-stream vegetation, biodiversity, and also ecosystem function. So, in-stream vegetation is a critical but often overlooked value in Melbourne's waterways. It's important because it provides habitat and refuge for in-stream biota. It engineers biogeomorphic processes. It increases hydraulic complexity. It influences sediment and chemical fluxes. It also influences food web energy pathways via primary production, and it also provides aesthetic values. Okay, so this conceptual model here shows visually the processes and interactions we seek to investigate in this research. So if we just look at the two shaded boxes labelled Q1 and Q2 at the top of the diagram, we can see that we're interested in getting a better understanding of how various flow metrics, for example, mean annual flow or FRE7 or FRE7, directly influence organic matter retention, storage, export, also coarse sediment yield, propagule retention, and in-stream vegetation growth and physical form. But then we're also interested in the feedback cycles that are generated between each of these variables through time. So we're looking at quite a lot of things. The primary aims of the research are to develop key in-stream vegetation species habitat preference curves based on velocity, depth, and substrate type. We're also looking to identify key life history stages from propagule retention to established vegetation that limit in-stream plant recruitment in streams under anthropogenic stress. Thirdly, we're interested in identifying characteristics of the flow regime that explain limitations to in-stream vegetation establishment at the different life history stages. And finally, looking to identify linkages between flow and in-stream vegetation in streams across the Melbourne water region with varying catchment characteristics and anthropogenic impacts. So the benefits of this research for Melbourne water are numerous. They include improved conceptual models linking flow, retention, and in-stream vegetation values. And these models are critical and underpin the Healthy Waterways strategy. This research will also develop and update flow guidelines, which ensure the protection and establishment of in-stream vegetation in urbanising areas. And it will also potentially improve prioritisation of management actions, which protect and improve both existing and new in-stream vegetation. Okay, so all of this research will be achieved through a large-scale stream survey across the Melbourne water region involving approximately 50 streams, where the sites will be randomly selected from a range of Melbourne water gauged and also unregulated streams to ensure a range across a number of variables, including things like directly connected imperviousness, mean annual discharge, FRE7, the number of zero flow days, distance to upstream barriers, etc. We'll use a line intercept survey, which will record the linear distance of substrata, retention features, so things like boulders, logs, or emergent vegetation, retained vegetative fragments or other CPOM, and also the retention feature nearest to any standing vegetation. In addition to this, we'll also collect sediments for a seedling emergence trial, and to measure sediment particle size and organic matter content. We'll also undertake a series of nursery and lab-based experiments using stream sediments that were collected from those surveys. And they'll be exposed to a range of different flow velocities and depth treatments in order to assess how varying flow regimes influence vegetation germination, colonisation, and species diversity. And finally, we'll also use some flumes to further identify flow velocity thresholds at which seedlings can be damaged and also uprooted. Okay, so to date we've undertaken a literature review. We've also spent a lot of time identifying methods for the field surveys and also the nursery and flume work. We've also started a pilot study. Well, actually, it's starting this week, so today. In the following year, we'll be looking to undertake the large-scale stream surveys using the line intercept survey method that I outlined before. And then we'll also be looking to assess vegetation emergence from the collected sediment samples. So we've got a lot of work to do, but we're getting through it, and the following year should bring some interesting results. So if you've got any questions on the project or any queries, feel free to drop me an email. My details are here up on the slide, or even give me a call. Yeah, great. I hope this was an interesting talk. Cheers.